Good afternoon, everyone. First, I would like to join my colleagues in thanking the organizers for the wonderful conference and the wonderful job they've done with the conference. And I'm really happy to be here. My talk will be about the genetics of the Vikings. It's a bit different than what you've heard so far, but it was part of my PhD project conducted at the University of Copenhagen. And as the title suggests, we try to reconstruct the genetic history of the Vikings using ancient DNA. First, I will have a general overview of ancient DNA and Vikings, and I will switch to the samples and the main results that we got. So what is ancient DNA? We've heard probably about it recently very often, but what is it? Well, it's basically any DNA extracted from dead organisms. And these are just some of the samples that we used to reconstruct ancient population histories, demographic events, and, for example, museum sample skeletons, cores, sediment cores, ice cores, chewing gums, etc. And the main issue with ancient DNA is, of course, contamination. And it happens when archaeologists or students touch the bones, for example, and transfer their own DNA to the bones. And this is well known. And therefore, present excavation is not a really big issue because people are aware of this and wear protective gloves and equipment, but this is still a major issue for older excavations. Another major issue is actually DNA fragmentation. Well, the DNA itself, the modern DNA itself, is a really long polymer with many so-called bases, A, C, G, and T. This is basically the DNA sequence, and it can be thousands of millions of these letters attached to each other. But the ancient DNA has these damaged sites, which basically fragments the DNA into many smaller pieces. So instead of this longer thread, we actually have this picture. And this is a screenshot from real DNA fragments. They're usually 30, 40, 50 base pairs in length. And the issue is to reconstruct them afterwards. This fragmentation has an exponential decaying pattern. And these are the fragment lengths of DNA. And as you can see, after a certain amount of time, the bulk majority of the DNA fragments is actually in the shorter range, less than 50. And the longer ones are essentially gone. And the further back we go in time, these profile will become very sharp, and this will be very short. Let's say if we go to Paleolithic or Mesolithic. Here I just show the major factors affecting this. The higher you have these factors, temperature, humidity, soil acidity, the faster this fragmentation will happen, and the decay will happen much faster. And yes, the issue with ancient DNA, as I said, is to actually reconstruct the original long sequence based on these smaller fragments. And one way of doing it is this is actually not as we do in reality, just to show more visually, but we find the overlaps of all the sequences basically to reconstruct the original sequence. So we find the overlaps where all these sequences match, and then again get the original longer sequence. After this, all the methods and approaches that we use is very similar to actually modern DNA. As soon as we reconstructed it, it's very similar. So. Very briefly about these guys, and I'm sure they don't need introduction, at least in this room for sure. But just to sum up, they changed the geopolitical, cultural landscape in Europe 1,000 years ago, but genetically speaking, we didn't know much about them, where they came from, what they were structuring in terms of genetics. This is a hilariously oversimplified version of the Viking World, of course, in terms of the arrows and everything, but this indicates the homeland Scandinavia. Again, these borders, as with temper, this is also geographically, there were never certain borders, of course. But it also highlights some of the major areas the Vikings settled, raided, and traded, etc. But the important part is all the green dots that you can hopefully see on the map. These are all the sites that we have samples from. And in total, we got 442 samples, most of them Viking Age, though we also had a bit early and later periods. But this covers pretty much all of the Northern Europe and Viking World, from all the way in the west, Greenland, and modern-day, present-day UK, Ukraine, and Russia in the east here. Yeah, about the samples. So I'll just give you an overview of some of the sites that we had samples from, but there were many. But for example, this is Varnhem in southern Sweden. This is a Christian burial site. We see the outline of the church and a lot of burials here. This one, on the other hand, is a boat burial on the Isle of Man. It's the Balladul site of just one individual buried in there. You've seen this one before, a very cool site. And the only thing you've seen in different versions, but the only thing is this yellow pipe that goes through it. That never changes. And the other one is this one. Yeah, I think Tim also very briefly mentioned this at the end of his talk, but this is at southern England. This is this so-called headless Vikings mass grave. And as you can see, it's 50 individuals in total, and the skulls are piled up on one corner, and the rest of the bodies are somewhere else. And so just to give an overall context of where we got the samples from. And after this, of course, with the permissions, because the sampling is destructive, with the permissions from the museums, we got the samples. Just to mention, yes, the teeth and the petrous bones are the best for ancient DNA. They preserve the DNA the best, and the petrous bone is at the base of the skull, indicated in the red color here. So that's the strongest part in the human body, and therefore preserves the DNA the best. And after that, it's the regular ancient DNA sort of workflow. Extraction, library building, sequencing, spending a lot of money. And then in this case, we got 64 billion DNA sequences that we had to analyze for this specific project. Now I'll get into the main results that we had. Yeah, for example, I'll start with the kinship analysis of some of the sites. A lot of samples were from larger cemeteries, so we would expect and we had actually a lot of related individuals, but I just chose a couple, probably most interesting ones to present here. For example, all these individuals, this is a church site in Faroe Islands, medieval site. All these individuals are actually one way or another related, could be siblings, cousins, but just for simplicity, I just focus on these 4 individuals. Those 2 little babies or even newborns are brothers, indicated here, first degree related, and the adults here were actually not related to each other, but they were second degree related to the baby. So the most probable explanation is that they're actually grandparents of these little boys. This one you heard yesterday, yes, the 4 brothers from Salme 2 burial. We, of course, don't have the parents, but actually one of these individuals, I don't remember, also had a third degree related from the sheep burial. And this, of course, emphasizes, that they went with big family groups. To be honest, maybe in retrospect, this wasn't very. Well, of course, this is cool result to find out, but this was not very unexpected. Given, of course, back in the days, you would go with people you trust or only people that would follow you. So in that sense, it was not really unexpected. But what is actually to find 2 individuals 500 kilometers apart from each other, one in present-day Denmark, the other one in Oxford in Britain. And there were 2 males, second degree related, so it could be, for example, grandparent, grandson, grandfather, grandson, or uncle and nephew. And I didn't show it here, but we also found maybe cousins, one in Uppsala, another one in Åland, again, hundreds of kilometers apart. So this was something, yes, we didn't expect. So I'll get into more nerdy genetic parts, but bear with me, I removed all the very narrow genetic pictures. So here we have each dot is a Viking individual on this map. And so we have about 400 of them grouped in based on their geography. But the important thing is that they basically don't form any clustering. They all form like a cloud here in the middle. And for comparison, we have more ancient populations like hunter-gatherers, farmers, and the Yamnaya here. And we wanted to see what is their relationship with ancient populations. And they're basically in the middle, meaning that they might have contributions from all these 3 groups. And indeed, that's what we find in this slide. this is what we call admixture analysis. These are all 400 Vikings grouped based on their geographical origin. And what I want you to take from this graph is just that they're all very similar and they all have these 3 colors basically. And the blue color, yes, each bar is a single individual. For example, in Salme, we have about 30 individuals, 30 bars. And yeah, they all have this blue, which comes from the Yamnaya. These are the Yamnaya from here. These are the comparative sets. The red one is the Mesolithic hunter-gatherers, the local ones. And the green ones are the Neolithic farmers that came during the Neolithic times. And just to focus slightly bit on Salme, we can see that all of the individuals have relatively similar proportions of blue, red, green compared to each other, for example, as comparison with different, and I'll get into that. That means they're relatively more homogeneous. They have very similar proportions between the individuals. And this is what we essentially see here. This is the genetic diversity in Scandinavia during the Viking Age. So here again, every dot is a single individual, and the further apart they are on the genetic plot, the more diverse they are genetically, more distant they are genetically. So the more spread out the samples, the more diverse it is, just to visualize. And of course, the winner is, in this case, Gotland, where we have the most genetic diversity. But all the other southern Scandinavian sites are also relatively diverse. And of course, here stands out Salme, which is relatively very homogeneous. They're not genetically very diverse, meaning that, again, they probably came from a very relatively close geographical region, well, as we know from the Lake Mälaren. And probably even more interesting was Norway. So here I just, with the arrows, show through different parts of Norway, but this is actually all 15 sites from Norway, and these are about 30 individuals. So this covers the whole of Norway. And what's interesting is the genetic diversity in all of Norway is actually less than in one site from Åland or Skara or Gotland for that matter. So yeah, this was definitely unexpected to find the whole this area geographically far bigger than a single cemetery, but genetically far less diverse. And in retrospect, of course, when we think about it, it's probably because the most activity was happening in this region during the Viking Age, where you had all the trade, maybe slave trade or other things happening. Basically, this was the most active region during during the Viking Age in Scandinavia. Then we tried to use more sensitive methods to actually see whether, well, in general, we couldn't see any obvious differences, but with more sensitive methods, maybe we could have actually detected. And yes, it's not super obvious, but we managed to get some noticeable differences. In this case, again, the colored ones are Vikings, Viking groups. And here we show Norway, Sweden, Poland, Denmark, UK, Italy, and Finland, modern populations from Europe as comparison. And in this case, already we see, for example, the green ones are the Swedish Vikings. They're relatively close to Sweden. The yellow ones are the Danish Vikings. They're close to present-day Denmark. The red ones are actually Norwegian Vikings closer to Norway, etc. And the blue ones are North Atlantic ones, Iceland, Greenland, Ireland. And they are closer to both UK and Norway, probably indicating the link, the fact that those Vikings were a mix of these two populations that we know from historical records. Well, based on this new data, we could ask a few other questions. For example, where do we see these three Scandinavian and North Atlantic, these 4 ancestries in the Viking world? Meaning, for example, during the Viking Age, where do we see this North Atlantic genetic component? And the North Atlantic is aka British component. And it was mostly in Britain, not very surprising, but also in Iceland and slightly bit in Greenland, but also in Norway. Here we ask where is the most Danish, for example, genetic ancestry, and of course it's in Denmark, but also in UK and a bit of in Scandinavia as well, but not in Greenland, for example. Here's the interesting part, probably the eastern ones. These are actually the Salme individuals, but also the other samples that we got from Sigtuna or Uppsala, near Uppsala area. So the genetic components, Swedish-like genetic components, are dominated in the east. So we don't find any in the Greenland or Iceland. And vice versa, the Norwegian component is very dominated in Norway, of course, but also in Iceland and Greenland. Again, this confirms, in terms of genetics, all the migration routes that we know from history and archaeology. And this is a good thing, I guess, that it matches. Then, because I've skipped many very nerdy genetic details to get to this, but I'll just mention that we've done a lot of analysis, and we showed that during the Viking Age we see this gene flow from the southern regions and eastern regions of Scandinavia into Scandinavia. So there are components that we don't see during the Iron Age and early Viking Age in Scandinavia, but then in later period during the Viking Age, we see these components coming first in Denmark and then kind of going up. So we see this gene flow into Scandinavia. So since we had whole genome sequence, we could ask some phenotypic traits. For example, if Vikings were lactose tolerant. And we had compared them with Bronze Age about 3,000 years ago. Essentially, the mutation that renders people lactose tolerant was 0, or very near to 0. So Bronze Age Scandinavians probably couldn't drink milk. In contrast, Viking Age was pretty high, and it was about 70%. So the frequency of these allele that rendered them lactose tolerant. And the interesting thing is that this number is very close to present day 80% roughly, which is the Northern European frequency for these allele that allows people to consume milk. Yes, we also were able to find out some pigmentation phenotypes, for example, hair and eye color, which allowed, for example, to reconstruct some of individuals, together with, of course, anthropology and osteology. But for example, this is one of my favorite cases where we also have the name of the individual. This is again from Varnhem, and the name of this lady is Kara, based on her gravestone, written in runes. But we also were able to find her hair color, eye color, and the museum did, I think, a great job reconstructing her, I think, entire body. And this is the same site in Faroe Island that I showed earlier, the grandparents and the grandchildren. But the grandparents actually had very well preserved skulls, so we were able, again, based on the genetic data, identify the hair color, eye color, skin color for these grandparents. And this probably means that they were also probably married. And this is just to emphasize that genetics can be very informative, together with archaeology and anthropology, to reconstruct, for example, a small history of a family, how they looked, how they were related. And very briefly about pathogens, because we shotgun sequenced the data, meaning that we were sequencing everything present in the bones, not just the human DNA, we were able to extract and sequence everything else, including the microbial DNA or the plant DNA or everything else connected with those bones. And we found whole list of, for example, viral pathogens. I'll just show some interesting ones here. Hepatitis B, herpes virus, and a long list of bacterial, such as Yersinia pestis, the plague bacteria, and lepra. And this is just a screenshot from an already published paper about parvovirus B19. I just wanted to show here because it clearly shows, for example, this individual, VK6 in Greenland, that was positive for parvovirus B19, and just emphasizes, for me at least, clearly the importance of these long-range migrations of the Vikings in transmitting DNA, the diseases back 1,000 years ago. So with this, perfect 20 minutes. Yeah, I won't go through the summary, but I wanted to thank you, of course, for listening, and the whole team of co-authors that made this paper possible. It was finally published in 2020. And of course, the funding bodies, because it was fairly expensive project, but thank you. Thank you.
Thank you, Ashot. Are there any questions? Kas on küsimusi? Ma viskan sinna selle.
I can speak in English. I presume that most of your sample were men, right?
Yes.
Okay. And I also presume that they were tilted quite heavily towards being elite. So in other words, they are not a cross-section of the society at that time.
We don't know.
You don't know? Okay. But did you discuss the probability of it that they are more tilted towards the elite and how all of this may influence your results and, for example, the comparison with modern populations and so on? And the second question, so this gene inflow from other parts of Europe, so the Eastern European and the in the, I don't know, what was it, Brittany and so on. Yeah. So are they slaves, or how did they end up there? So what are the reasons for that?
Yeah, thank you for the questions. So the first one with the elite individual. So in this paper, when we used Vikings, it was an umbrella term for Viking Age Scandinavians, or if they're outside Scandinavians, if they had any connection, maybe archaeological or historical connections with Scandinavians. So we don't see any Viking-specific activity, well, apart, of course, from Salme, for example. But the rest were just Viking Age. If that individual was from Scandinavia during Viking Age, we just sampled them. So we don't know about any status or these things. So in that case, that's why we wanted to be very careful saying Viking Age Scandinavia as opposed to usually Vikings themselves that were actually raiding, and just whoever was present at that time during the Viking Age in Scandinavia. So yeah, we can't distinguish. I mean, of course, well, the data is out there. So, if there is a specific individual with maybe king goods or things like that, of course, we can always go back and try to see how they look. But this was more general overview of how the Vikings were in terms of genetics. And regarding the second question, the southern and eastern migrations that we see it could be, of course, slaves, but also could be traders that coming in. We can, of course, only speculate. We see genetically that there's this new genetic component that are coming during the Viking Age into Scandinavia that was absent during Iron Age and early Viking Age. So, and we can only speculate again that it's probably merchants coming in, for example, to trade and maybe dying there. Of course, a lot of probably slaves were brought back with the Vikings themselves. So, yeah, it could be these factors, of course, and probably more others. Yeah, unfortunately, it's like, there's no clear answers to that.
Thank you. I can comment on actually on the elite burial question, because predominantly the Viking Age burials are cremations, and you cannot do ancient DNA on those. But from particular geographical regions, there are inhumations, like Gotland, for example. Yeah. So in terms of that, there is a possibility that there is a wider variety of people. But from the Viking Age proper, we lack a lot of graves, so not all people were buried. And most likely we lack people from the lowest social strata. But when we come into the very early medieval period and the Sigtuna samples, these are samples taken from very early medieval cemeteries. And there you have people from all social levels, and also men, women, and children of different ages. So there I think we have a better representation of people from different social groups. But yes, it's problematic in terms of most burials not being in inhumations, and all the ones that are inhumations from particular time periods are others in a sense, because they've done it in a different way.
Just to get back to the Salma ship and your results that you published there, it's a tight-knitted community on the boat which have can trace their DNA back to Swedish areas. What would your expectation be of a local community within the proximity of this burial, when I think you said that there was very much a Swedish DNA presence sweeping across the eastern Baltic? Just devil's advocate, what would you think? What would you expect?
Well, we would expect the locals would be genetically different. For example, if we don't have comparative data set from Estonia or Baltic region during the Viking Age. I'm sure there will be more data coming out to compare them. But we don't have any direct comparison with the local Eastern Baltic areas with the Vikings and especially the Salme and Swedish samples. But we would definitely be able to, so they would definitely be more distant than, for example, Danes and Swedes or Swedes and Norwegians. So we should be able to detect these differences more clearly than within Scandinavia. Yeah.
Thank you.
I think we'll, thank you.